Bismillahirrahmanirrahim. Assalamu alaikum, dear students. I hope that all of you will be fine and in a good state of health. I am Dr. Muhammad Ali Hashmi from University of Education at a campus, and we are here to study the subject of Introduction to Biochemistry. This is lecture number 24, and it's about significance of lipids in membrane transport mechanisms. This is the part 2 of the previous lecture. So in the previous one, we studied about the different transport mechanisms. In that one, we discussed about the passive transport, and in that passive transport, we discussed the simple diffusion. Then there was the facilitated diffusion and how that operates with the help of those carrier proteins. Simple diffusion just operates by diffusing the molecules from an area of higher concentration to the area of lower concentration, while the facilitated diffusions that was with the help of specific protein channels or carrier proteins. So protein channels help move the polar molecules or other substances that have problems passing through the lipid bilayer, and certain carrier proteins are also there which can bind to molecules, and then they can take that molecule inside the cell and then release that molecule in there. So all these processes of simple passive transport, they do not require energy, or they do not use the energy of the cell. So in this next part of the series, we study about the significance of lipids in the different transport mechanisms, which will include now active transport, and then endocytosis and exocytosis. So active transport will be the transport which will be against the concentration gradient, so it will be using some of the cell's energy as well. And then we have mechanisms in place to move the larger molecules inside and outside the cell. So those mechanisms will be studied in the endocytosis and exocytosis. First, we come to the active transport. Active transport mechanisms require the use of cell's energy, and that energy is usually in the form of adenosine triphosphate. So you know that adenosine triphosphate is your energy store or energy reserve molecule. So wherever energy is needed, these molecules go there, hydrolyze, and provide energy to the body for its functions. If a substance must move into the cell against its concentration gradient, that is, if the concentration of the substance inside the cell is greater than its concentration in the extracellular fluid, and vice versa, the cell must use energy to move the substance. So you know that pushing anything against its concentration gradient, that requires energy. So just take an example that if water is flowing from a higher place to the lower place, it just flows without any input or without the use of any energy. But what if you have to take water from the ground and to the water tank on your roof? So that is definitely against the concentration gradient, and you will have to use the energy. So where will that energy come from? So if you are using a water pump, you use the electrical energy that is converted into mechanical energy in the pump, and it takes the water from the ground and pushes it against its concentration gradient to the rooftop. So that requires energy because we are moving something against its concentration gradient. So whenever you move something against a concentration gradient, that will require energy. So that's the case with the active transport in the cell, that when we move some substances against their concentration gradient, we use the energy in the form of adenosine triphosphate. So some active transport mechanisms move small molecular weight materials such as ions through the membrane. So there are some mechanisms which will only move small molecular weight materials like ions, so sodium ions, potassium ions, etc. But there will be other mechanisms which move much larger molecules as well. So if you look at sugars and other similar carbohydrates and these molecules, they are much larger in size than those ions we just discussed. They are also moved through the active transport process. So the primary active transport functions to transport sodium and potassium actively. Actively mean against the concentration gradient, mean with the use of energy. One of the most important pumps in animal cells is the sodium-potassium pump. There is a pump in all animal cells which is called sodium-potassium pump. They say sodium-potassium ATPase is also its name, which maintains the correct concentrations of sodium and potassium in living cells. So what is the function of that pump? It maintains the correct concentration of these ions in the living cell. So if the concentration of sodium becomes higher in the cell, it is transported outside the cell using these sodium potassium pumps. So even in the case the concentration of sodium potassium pump is higher outside the concentration of sodium is higher outside the cell, still it will be moved out from the cell because it is not needed in the cell anymore. So that will push it against its concentration gradient, and it is not possible without the use of energy, and that pump is responsible to use the energy and move these ions outside the cell. So sodium potassium pump moves potassium into the cell while moving sodium out at the same time at a ratio of 3 sodium for every 2 potassium ions moved in. So this is a mechanism that whenever 3 sodium ions are moved outside the cell, 2 potassium ions are moved inside the cell as well. So these things happen at the same time. Sodium is a small ion, potassium is a bigger ion than sodium due to having one extra shell. So whenever 3 sodium ions are moved outside the cell, 2 potassium are also moved inside the cell at the same time. The sodium potassium ATPase pump exists in 2 forms depending on its orientation to the interior or exterior of the cell and its affinity for either sodium or potassium ions. So this pump exists in 2 different forms. And what are those 2 different forms? They depend on its orientation. Either it is oriented to the interior of the cell or it is oriented to the exterior of the cell. So that is one criterion for its 2 forms, and then the second criterion is the affinity of it for either sodium or potassium ions. So either it is affinitive to the potassium ions or sodium ions that also give it a different form. So the whole process of the movement of these ions across the cell membrane, it consists of the following 6 steps. So the first step is with the enzyme oriented towards the interior of the cell. So now the sodium potassium pump is oriented towards the interior of the cell, mean towards the cytoplasm. So the carrier has a high affinity for sodium ions. Now when it is oriented inside the cell, it has high affinity for sodium ions. So 3 ions bind to the protein. When it has a high affinity for sodium, 3 of the sodium ions, they bind to this carrier protein, the sodium potassium pump. So next step is the ATP is hydrolyzed by the protein carrier and a low energy phosphate group attaches to it. So now an ATP molecule is hydrolyzed to provide the energy to this protein carrier or the pump, and a low energy phosphate group attaches to this pump. You can see here that when this pump is oriented towards the inside of the cell, this is cytoplasm and this is extracellular space. So this is inside the cell and this is outside the cell. The pump is oriented towards the inside of the cell, and 3 sodium ions bind to it. Then an ATP molecule is hydrolyzed and a small phosphate molecule gets attached to this pump. And what happens at number 3? As a result, the carrier changes shape and reorients itself towards the exterior of the membrane. Now what happens? That it changes its shape. You can see that it was oriented towards the inside of the cell. Now after the use of ATP, it has changed its shape and it is oriented to the outside of the cell now. The protein's affinity for sodium decreases. Now when it is oriented towards the outside of the cell, its affinity for sodium decreases and its affinity for potassium increases. So what happens that it just throws away those 3 sodium ions towards the outside the cell, because its affinity for those ions has now decreased. The fourth step is the shape change. That shape change increases the carrier's affinity for potassium ions. Now its affinity for potassium ions is increased, and that is why 2 such ions attach to the protein. So because of the bigger size, 3 potassium ions cannot be attached. 2 of them, they get attached to the protein or this sodium potassium pump. So what happens subsequently, the low energy phosphate group detaches from the carrier. Now when the potassium ions, they get attached to the protein, the low energy phosphate group that was attached to the protein on the inside of the cell to release the ATP, that phosphate group detaches from there. And next, the phosphate group removes and the potassium ion get attached. Now the carrier protein repositions itself again towards the interior of the cell. So now from outside of the cell, the protein now reorients itself to the inside of the cell. And now the carrier protein now in its new configuration has a decreased affinity for potassium, and the 2 ions are released into the cytoplasm. So now when the protein is oriented towards the inside of the cell, it has a decreased affinity for potassium, and the ions which were attached to it, they are released into the cytoplasm. And again, when the ions are released, now the protein has a higher affinity for sodium ions again, and the process starts again. So you can see here that after releasing sodium, 2 potassium ions got attached to the protein, and then the phosphate group gets removed from here. The protein reorients itself, and now the potassium ions are released inside the cytoplasm. When the potassium ions are released inside the cytoplasm, it has again higher affinity for sodium ions, so it will the process will restart here. Now let's watch a little animation for understanding this in a visual way that will increase your understanding about the process, how it happens.
Some ions, such as sodium and potassium, require transport against their concentration gradients. The sodium-potassium pump actively transports these ions from where they are less concentrated to where they are more concentrated. This process requires energy from the cell and is known as active transport. Energy from ATP is used to move 3 sodium ions shown in red, out of the cell, and 2 potassium ions, shown in blue, into the cell. Because the pump uses ATP directly, this type of transport is called primary active transport. Another type of active transport, secondary active transport, also moves chemicals against gradients. It doesn't use ATP as a direct source of energy. Instead, it uses the downhill gradient from one ion or molecule to drive the uphill movement of another ion or molecule. For example, sodium and glucose move into the cell by secondary active transport in a process called cotransport or symport, because both substances are moving in the same direction. Sodium ions move down their concentration gradient created by the sodium-potassium pump, but because glucose concentrations inside the cell can quickly exceed those outside the cell, glucose moves against its concentration gradient.
Yeah, now you have seen that the sodium-potassium pumps pump the sodium and potassium outside and inside the cell, and that was the primary active transport because it uses energy directly from the ATP. There is a secondary active transport as well, which uses these coproteins, and it transports sodium ions and the glucose ions inside the cell. So sodium ions are transported towards their concentration gradient from an area of higher concentration to the area of lower concentration, but glucose in this case is transported against the concentration gradient because inside the cell is a small place and the glucose concentration is higher inside the cell than it's outside the cell. But that glucose is transported using these coproteins, which move the glucose molecules along with the sodium ions, and the movement is by using an active mechanism which is called secondary active transport. So in addition to moving these small molecules and ions through the membrane, cells also need to remove and take in larger molecules and particles. So some cells are even capable of engulfing entire unicellular microorganisms. The uptake and release of larger particles by the cell requires energy. A large particle, however, cannot pass through the membrane even with energy supplied by the cell. So what happens that these cells sometimes they engulf the whole unicellular microorganism, in that there is a very large particle compared to the cell. Compared to the size of the eukaryotic cell, it is engulfing a very large particle, and that just does it through these mechanisms which we are going to study now. And these large particles, they won't be able to pass through these carrier proteins or through the plasma membrane in a simple way. So they have different mechanisms of transport. So these are two primary mechanisms which are called endocytosis and exocytosis. Endocytosis is used to take in the materials inside the cell, and exocytosis will be used as to move the materials outside the cell. The first one is endocytosis. Endocytosis is a type of active transport that moves molecules, that moves particles such as large molecules, parts of cells, and even whole cells into a cell. So endocytosis is a bigger scale movement. It moves the larger molecules, very large molecules, and sometimes parts of the cell, and sometimes even a whole cell into the cell. So there are different variations of endocytosis, but all share a common characteristic. What is that common characteristic? The plasma membrane of the cell invaginates, forming a pocket around the target particle. So what happens in the endocytosis, that plasma membrane just invaginates, it creates a little cavity in it, and then it wraps around the particle which it needs to take inside. It just forms a pocket around it, and then pocket pinches off, resulting in the particle being contained in a newly created intracellular vesicle. And then that portion pinches off around it, and the particle gets enclosed in a membrane, in a little newly created membrane or a vesicle formed by the plasma membrane. You'll clear yourself about this process on the next slides, and with the help of some figures. But first we see that how many types of endocytosis can be there. So endocytosis can be of the following three types. It can be phagocytosis, it can be pinocytosis, or it can be receptor-mediated endocytosis. So these three different types of endocytosis will be discussed in the next slides. So first is phagocytosis. What is phagocytosis? The condition of cell eating is the process. Phagocytosis means the condition of cell eating. It is the process by which large particles such as cells or relatively large particles are taken in by a cell. So in phagocytosis we take in the larger particles into the cell, and in preparation for phagocytosis a portion of the inward facing surface of the plasma membrane becomes coated with a protein called clathrin, which stabilizes this section of the membrane. So the portion of the plasma membrane which is forming an invagination towards the inside, it is coated with a protein called clathrin. That protein actually stabilizes this section of the membrane and helps it so that it does not burst or get ruptured from here. And the coated portion of the membrane then extends from the body of the cells and surrounds the particle, eventually enclosing it. So here you will see that this is the invagination I was talking about. This is the membrane, and when a particle comes in contact with it, it just forms an invagination around it, and there is a protein called clathrin which helps stabilize this plasma membrane so it does not get ruptured. And then this portion just covers this whole particle and encloses it into a vesicle. That vesicle is moved inside the cell. Once the vesicle containing the particle is enclosed within the cell, the clathrin disengages from the membrane and the vesicle merges with a lysosome for the breakdown of the material in the newly formed compartment, which is called this endosome. So what happens that this vesicle moves inside. Now the clathrin protein just disengages because its function is finished, and this vesicle will go inside the cell and bind to a lysosome. So what are lysosomes? Lysosomes have the enzymes to break down these different particles. So lysosome releases those enzymes and starts the breakdown of this particle which just entered the cell. Next is pinocytosis. So it is also a variation of endocytosis, and it literally means cell drinking. Phagocytosis was cell eating, and pinocytosis is about cell drinking, and it was named at a time when the assumption was that the cell was purposefully taking in extracellular fluid. In reality, this is a process that takes in molecules, including water, which the cell needs from the extracellular fluid. So through the pinocytosis, cell takes in the liquid things, including water. In phagocytosis, the cell takes in the solid particles, but in pinocytosis, cell takes in liquid molecules. So pinocytosis results in a much smaller vesicle than does phagocytosis, and the vesicle does not need to merge with the lysosome. So because it's liquid, it does not need to merge with the lysosome to get broken down when it reaches inside the cell. So this pinocytosis vesicle, when reaches inside the cell, it just releases the contents. So pinocytosis is used to bring small molecules into the cell and to transport these molecules through the cell for their release on the other side of the cell. So you can see that these are small liquid molecules which just come in contact with the membrane, and then membrane makes an invagination around it and close it into a vesicle and then releases that vesicle into the cell. So that is how these liquid molecules are transported into the cell. The next one is receptor-mediated endocytosis. So a targeted variation of endocytosis employs receptor proteins in the plasma membrane that have a specific binding affinity for certain substances. So there are some certain receptor proteins, you can see here the green ones, which have specific affinity to bind with certain substances. Like this protein has bound a substance with it. So those substances bind to these proteins, and then the process of endocytosis happens. So in receptor-mediated endocytosis, as in phagocytosis, clathrin is attached to the cytoplasmic side of the plasma membrane so that it stabilizes the plasma membrane. So these receptors, they mediate the endocytosis. That is why this is called receptor-mediated endocytosis. If the uptake of a compound is dependent on receptor-mediated endocytosis and the process is ineffective, the material will not be removed from the tissue fluids or blood. So if this process does not get affected, it is not effective and it cannot function properly, the material will not be removed from the tissues. It will still keep there, and it will stay in those fluids and increase its concentration. And some human diseases are caused by the failure of receptor-mediated endocytosis. So for example, the tissues or the cell has to release these substances outside the cell, and due to some malfunction, the receptor-mediated endocytosis cannot work properly. That will cause a disease due to that, because the concentration of that substance will become higher and higher and it won't be transported outside. For example, the form of cholesterol, the term low density lipoprotein or LDL, that is also referred to as bad cholesterol. It is removed from the blood by receptor-mediated endocytosis. So bad cholesterol is removed from the blood through this endocytosis. But in the human genetic disease, familial hypercholesterolemia, in this disease, the LDL receptors are defective or missing entirely. What happens in this disease, that these receptors which bind LDL and transport it outside, they get missing or they get defective. So people with this condition have life-threatening levels of cholesterol in their blood. So that increases your blood cholesterol levels, and you have the disease then, because your cells cannot clear LDL particles from the blood. Next is exocytosis. It is the reverse process of moving materials into a cell. So exocytosis is the opposite of the processes discussed on the previous slides, in that its purpose is to expel material from the cell into the extracellular fluid. So waste material is enveloped in a membrane and fuses with the interior of the plasma membrane. So it happens the same way but from inside the cell. So this fusion opens the membrane envelopes on the exterior of the cell, and the waste material is expelled into the extracellular space. So we'll watch a short animation to summarize and understand these processes of endocytosis and exocytosis, so you have a more clear picture of how these things happen.
The substances taken in by single-celled organisms are often particles or large polar molecules that cannot cross the hydrophobic plasma membrane. Many single-celled eukaryotes employ endocytosis to ingest such food particles. In this process, the plasma membrane extends outward and surrounds the food particle. Cells use three major types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterium or a fragment of organic matter, The process is called phagocytosis. If the material the cell takes in is liquid, it is called pinocytosis. Specific molecules, such as low-density lipoproteins, LDL, are often transported into eukaryotic cells through receptor-mediated endocytosis. Molecules to be transported first bind to specific receptors on the plasma membrane. The interior portion of the receptor protein is embedded in the membrane. The protein clathrin coats the inside of the membrane in the area of the pit. When an appropriate collection of molecules gathers in the coated pit, the pit deepens and seals off to form a coated vesicle which carries the molecules into the cell. Exocytosis is the reverse of endocytosis. This process results in the discharge of material from vesicles at the cell surface to the outside of the cell.
So that video animation summarized the endocytosis and exocytosis processes beautifully, and I hope that you have understood these mechanisms clearly now. Here is a little assignment or 2 questions for you, which you need to address after listening and understanding this lecture. So these will tell how good you have got the point. And with this lecture, we finish the chapter of lipids, and next we move on to the next chapter from the next lecture. Here is another little scenario for you, which you will solve. After reading the statement, you will choose any one of the correct option, and you can ask any questions from your teachers in the classroom and about these questions as well. But just give these questions a try yourself first, and then if you need to verify your answers, you can ask your teachers about them. The references for the preparation of these lectures are given here, and I wish you the best of luck for your studies and your exams. And if you have any questions, you can ask in the Google Classroom. Thank you very much for watching, and Allah Hafiz.